Yes, I muted. Thanks for letting me know. Yeah, I'll check if you paid attention. Kidding. So I'm asking, can this be the start codon of a gene and this the stop codon of that gene? So let's see, let's go by parts. One of them would be, are they in frame? So if this is the start codon, then I'd go like, okay, that's the first codon, then that's the second codon, third, fourth, fifth. And so it's not in frame because this TAA is not read together. This would actually be read ATA, and then the ACG is the next codon. You see that? So see, I don't know. I lost myself here. But you look at every three, and you see, does it happen that this TAA is in frame to be a stop codon? And there are some genetic diseases where if you delete or insert, remember there are indels, one or two nucleotides, the reading frame changes, and it induces to an early stop codon, and then a protein doesn't get made, and that is where you would see an effect on the phenotype. You'll see more on this later. But the two reasons why the gene would not be from here to here is that this TAA would not be read in frame. And when I say that, I mean look at every three nucleotides. You wouldn't read the TAA together if you did that. And I put them really close because it's difficult to keep track of this. Luckily, the computer does this for you when we are running the software predicting genes. And you might go like, why did you put this thing here? Oh, it's because I wanted you to have some idea of how large proteins are. Because if you have no idea, and if you ignored this thing that this is not in frame, you'd say, oh, I do have a protein that has 1, 2, 3, 4, 5, I don't know, 5 amino acids. That sounds reasonable. Actually, no, 5 amino acids would be way too short. And here is how often you would find something with 5 amino acids. Basically, well, never. Here is most proteins. I think, I don't remember for which organism this is, because this actually varies depending on the organism. Might have been human, might have been yeast, but it varies a little bit. So this guy, I'd say maybe 200. Maybe most proteins have about 200 amino acids, but nobody has only 5. See, you have to have at least, how many is this? I don't know, 50? Okay, that would be like a peptide. So this histogram here, how often you see some protein of these different lengths, was to try to help you that this was too short to be a gene.